Hello students, welcome. I am Jyoti Rajput and today we will study about the chromatin. As you can see this diagram, this is a nucleus and here this is called as chromatin. Actually it is a DNA. The DNA it is packaged in the form of chromatin. So here whenever the cell is in interphase, very clearly we will be able to observe the chromatin and then only the cell it does the mitosis and meiosis type of cell division. And there is more importance about chromatin that it also prevents the breakage of the chromosome. And in the chromatin phase only, here the gene expression is going to take place. It means the genes, here the flow of information will take place from gene to RNA, and then the RNA it will give rise to the protein. So here this is called as gene expression. And even DNA replication will take place when the DNA it is in the chromatin form. Here further when we talk about the chromatin, it is the most important part of the nucleus because here all the information which is present in the DNA in the chromatin form, it is the one which is going to transfer from one generation to the next generation. So here chromatin its function it is seen in the heredity. The chromatin it contains the DNA, it contains the RNA and it also contains the protein. Sometimes here the chromatin it is so much compact that the DNA sequence they are no more functional. It becomes inactive and it becomes inaccessible. That region is going to be called as heterochromatin. And here inaccessibility it is because of the binding proteins. Here the DNA it is along with the histone protein and when they are very much compact to each other, so that region is called as heterochromatin. कॉम्पैक्ट होने के वजह से बिकॉज ऑफ द मोर एंड मोर कॉम्पैक्टनेस देर आर वेरी लेस free DNA. There are very less loose DNA present, which is unable to do the transcription, which is unable to produce the RNA and protein here. So this DNA along with the histone protein, when they are in very compact stage, very compact form, here they are completely inactive or you can say inaccessible. So on the basis of this, the chromatin they exist in two forms. Here the very first is the euchromatin and the second one is the heterochromatin. Here when we see the chromatin, when it is stained by using a nuclear stain, if it is dark, then that region is called as heterochromatin. And here you can see the region which is dark, that region is received the more stain, that is why it is dark and it is called as heterochromatin. And the region which is light, where the stain it is very less, that region is called as euchromatin. So among them, the euchromatin is the one where DNA it is loosely arranged. That is why it is easily being transcribed. And here the DNA, the genes which is present, it will give rise to RNA and it will give rise to protein. When we talk about the heterochromatin, here the DNA along with histone protein, they are very compactly arranged. They are not loosely arranged, due to which the compaction, due to this the there is no free DNA and they are unable to do the transcription, and here there is no formation of RNA and nor formation of the protein. So here the euchromatin and the heterochromatin it can be distinguished depending on the staining properties and it is visible during the interphase. When we talk about the euchromatin, here you can see the loosely arranged DNA with the histone protein. They are loosely arranged. So loosely arranged होने के वजह से ज़्यादा enzymes और इस तरीके के proteins जो है वो attach होके transcription easily कर सकते हैं. So in the euchromatin, whenever we see, they are always lightly stained. Whenever we use a stain, nuclear stain, so it is lightly packed, it is the lightly packed chromatin. It contains the DNA, RNA, and protein. And here it is the one which is having the genes, and this is the active form of the chromatin. Here, euchromatin it comprises the most active portion of the gene. It means it is the one which is going to do the transcription. So, when we talk about the human genome, 92% of the human genome it consists of the euchromatin. Rest is heterochromatin. And whenever we see the here chromatin, the lightly stained, it is going to be called as euchromatin. And here it is less condensed, it means it is loosely arranged, so more free DNA with histone protein they are available. Here it is also called as dispersed chromatin because they are loosely arranged. And the structure of the euchromatin, it is reminiscent of an unfolded set of beads. So whenever we see the DNA, imagine this green strand, it is the DNA, and here these proteins are called as histone protein. So DNA with the histone protein, this structure is formed, that is called as nucleosome. This is called as nucleosome. So the structure of euchromatin is reminiscent of an unfolded set of beads along a string, where it appears like a bead represents the nucleosome. Here, when we observe the nucleosome, this is the histone protein and this is the DNA which is coiled around the histone protein. So they gave rise to a structure that is called as nucleosome. The nucleosome, here it consists of this histone protein and here the DNA. And here whenever we observe the DNA, the total base pairs in the DNA in this nucleosome is going to be 146 base pairs of DNA it is going to be present. So here 146 base pair of DNA it is coiled around this histone protein. And here in euchromatin this wrapping is loose so that the raw DNA it should be freely available so that the enzymes and protein they will attach, and now the transcription process will easily be takes place. Here, now we will study about the functions of the euchromatin. As you can see, this is a DNA. There are number of genes present in DNA. And here, as you can see, this DNA, it is freely available, loosely arranged. So, here, it is called as euchromatin. It is active, it means it is going to do the transcription process. The DNA it will convert into the mRNA, it means the information flow of information is going to be from DNA to the RNA. So here on this DNA there are going to be genes, and here some proteins and RNA polymerase enzyme, they are going to bind this DNA sequence and the transcription process is going to start. So we can say that not all euchromatin is necessarily transcribed, but in general that which is not transcribed into heterochromatin to protect the genes while they are not in use. So here there are some of the euchromatin also which is not transcribed, so it is protected whenever they are not in use. We can here see a video which will completely explain about the transcription process, that how the DNA transcription takes place in the euchromatin.
The DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA.
So in this video, it is completely explained that how the flow of information is taking place from the DNA to RNA, and then the RNA it will give rise to the protein. So this is called as the DNA it is transcribed. So here it takes place in the euchromatin. Now we will talk about the heterochromatin. The DNA with the histone protein, they are compactly arranged, due to which there is no loosely arranged DNA. The DNA is not freely available. So now the proteins and the RNA polymerase enzyme, it is unable to get attached, and here the transcription process, it is going to be prevented. So here heterochromatin, here you can see the darkly stained structures. These are heterochromatin, and here the DNA along with the histone protein, it is tightly packaged, and here dark stained part of the chromatin, it is the heterochromatin. They represent that the DNA, it is very in condensed state. So it is also seen in the chromosome also. In the chromosome also the condensed state condition, it is seen. And here this region, this chromatin part, it is always inactive because the DNA, it is very compactly arranged, and here because it is not free, so it is cannot be transcribed. Further, when we talk about the nucleolus which is present in the nucleus, there is also heterochromatin seen. So here, when you can see the perinucleolar chromatin, here this is the heterochromatin part. Here, when we see the intranucleolar chromatin, this is also the heterochromatin part, and they are joined to each other. Altogether, they are called as nucleolar chromatin. This is the example of the heterochromatin. Here, when we see the nucleus, here when we see, there are some clumps of darkly stained heterochromatin which occurs in close contact with the fibrous laminar. So whenever we see this region, this is the nuclear membrane, this is the nuclear membrane. In the border you can see there are some clumps, and here these clumps, these are the heterochromatin which are attached with the fibrous laminar, which was always in contact with the inner nuclear membrane. This is called as condensed peripheral chromatin. This is also an example of heterochromatin. Now we will talk about the functions of heterochromatin. Here, heterochromatin, it is always associated with number of functions like gene regulation. They are protecting the chromosome, and here because of the dense condition, because of more packaging, it is less accessible, so that the protein can be attached and the transcription process could takes place. Here, even here the heterochromatin, it is also the one which is protecting the DNA. So whenever we observe the DNA, here the DNA, it is a double stranded molecule. The DNA, it is a double stranded molecule. So here the ends of the DNA, it can be interpreted that it is a damaged or viral DNA. And here because of this, the cell cycle, it could stop. The cell cycle, it could be arrested. Here the DNA repair can start, the DNA destruction, it can also be start, and there may be condition that endonuclease which is present in bacteria, it can also damage these kinds of DNA. So that is why more and more packaging of the DNA, it is essential, and it is done by the heterochromatin. In this we can see some region of the chromatin are very less densely packed with fibers that display a condition comparable to that of chromosome at mitosis. So here heterochromatin, as these are the inactive one, they do not do any kind of transcription. They are generally clonally inherited. Here there is an epigenetic inheritance seen in the heterochromatin. It means the genes which are present in the heterochromatin, they are going to be the DNA sequence, they it remains unchanged, and here as it is, they are going to get transferred to the daughter cells during the mitosis or meiosis type of the cell division. So this is how all about the chromatin was, and here the video ends, and thank you very much.